Good morning, students. So in this class, it is a continuation part of reproduction in bacteria. So in the last class, you have studied the asexual methods of reproduction in bacteria, which will take place by binary fission and endospore formation. So here in this class, you will study about sexual reproduction in bacteria. Sexual reproduction in bacteria. So sexual reproduction in bacteria, it is also called as parasexuality or genetic recombination. So bacteria exhibit a primitive type of sexual reproduction, so which is called as either parasexuality or we can call it as genetic recombination. So here in this method we could find there will be the transfer of a genetic material, the genes or the DNA will be transferred, the transfer of genetic material from one bacterial cell to the another bacterial cell. Okay, so it is a primitive type of the method of reproduction seen in bacteria. As such, the sexual reproduction which is seen in case of the higher organism, so there is during the sexual reproduction there is no meiosis taking place, there is no formation and fusion of gametes seen in case of bacteria, but there is an involvement of two bacterial cell and there will be the transfer of the genetic material, the genes from one bacterial cell to the another bacterial cell. Okay, and a new genetic makeup is being seen in another bacterial cell, a recipient bacterial cell. So it is called as genetic recombination. So it is a primitive type of a reproduction. Okay, so in case of sexual reproduction, you know, two individuals are involved. So here also two bacterial cells. So it is called as parasexual. Okay, so sexual reproduction in case of bacteria is also known as parasexual or genetic recombination. Sexual reproduction in case of bacteria takes place by three steps. Takes place in three different ways: conjugation, transformation, and transduction. Genetic recombination, yes, takes place in three different methods. Okay, so that is three different methods in case of a sexual reproduction or genetic recombination: conjugation, transformation, and transduction. So one by one in detail. Okay, so you will study now. Conjugation. It was first explained by Lederberg and Tatum. Lederberg and Tatum. Conjugation was first explained by Lederberg and Tatum. So conjugation, it involves two bacterial cell. Okay, so here we could find two different forms of bacteria. We can call it as dimorphic. One is called as F plus bacterial cell. Another is called as F minus bacterial cell. So there are two bacterial cells. Two different forms of bacterial cells are being involved. It is F plus and F minus forms of bacterial cells. F plus bacterium is also known as donor. It is also being called as male bacterium. F minus is also called as recipient. Okay, so it is also called as recipient. So what are the characteristic features of F plus bacterium and F minus bacterium? F plus bacterium or a donor bacterium so in case of F plus bacterium, apart from genetic material, there is the plasmid present. Okay, what is plasmid? In the last class I have explained, apart from genetic material in the cytoplasm, there is an extra DNA present. Okay, so extra chromosomal DNA. Okay, so extra DNA which is being found in the cytoplasm, so that is called as the plasmid. On this plasmid has a gene for fertility. Okay, it has the F plus gene, fertility gene. What is this fertility F plasmid? Okay, so here we could find that gene is responsible for having the production of the pili. What are pili? Finger-like, hair-like projections produced by the bacterium like this. Okay, so you could find. So this plasmid has a gene for fertility, and that gene is responsible for production of the pili as well as transfer of the genetic material. Okay, transfer of genetic material from this bacterial cell to the another bacterial cell, from donor to the recipient. So these are the characteristic feature of the donor or F plus bacterium. Due to the presence of the gene for fertility on a plasmid in this bacterium, so that bacterium is called as F plus bacterium, or it is called as, also known as donor, or also sometimes called as male bacterium. Okay, and it has a pili. Okay, what are pili? Hair-like structure, hollow in nature. So another bacterium another bacterium bacterium, it is called as F minus or recipient. So here, what are the characteristic feature? In this bacterium, there is absence of a plasmid. Plasmid is absent. A gene for fertility is also absent. So it receives the genes from donor bacterium. So it is called as recipient. It will send the genes to the recipient bacterium. So it is called as the donor. Okay. So here in case of conjugation, there are the involvement of two forms of bacteria. One is called as F plus and another is called as F minus bacterium. Whenever these two forms of bacteria come close to each other, yes, F minus come close to the F plus bacterium. So this So when it comes close to the F plus bacterium, the F minus or recipient bacterium come closest. So the F plus bacterium has a 1 to 4 pili. That pili forms a bridge. It attaches by the help of the pili. It attaches to the recipient bacterium, and this forms a bridge-like structure, and it is called as conjugation bridge or called as conjugation tube. It is like a tubular structure, so it can also be called as conjugation tube. So, by the help of a pili, I have told function. It is used for means of attachment to the host or helps during the time of reproduction. So you can find here. So it helps in attachment to the recipient F minus bacterium and forms a bridge-like structure, tube-like structure. So it is called as conjugation bridge. Listen here, the plasmid which has a gene for fertility, and the plasmid, what is the important characteristic feature? It has a capacity to replicate on its own without depending on the genetic material. Okay, what is replication? Produces another copy of its own. So, there will be another copy produced like this. One more copy of plasmid is produced. After the replication of the plasmid takes place. So now what happens? See here, this slowly moves like this. Okay, moves into the what? And recipient bacterial cell. See here. So you can find here, by the help of conjugation bridge, there will be the replicated plasmid, a genetic material, the DNA will be transferred to the F minus bacterium. From F plus bacterium, it will be transferred to F minus bacterium. Yes, so now what happens? So when the genetic material is being completely transferred, so now the conjugation bridge will be broken down. So now this F plus bacterium has transferred the plasmid, a genetic material DNA, to the recipient bacterium. So now this F minus bacterium has been converted as F plus bacterium. Why? Because this bacterium also has the gene for fertility on the plasmid. So there is plasmid present. Okay, so this method of transfer of a genetic material by the help of a conjugation bridge, so that is called as a conjugation. So transfer of genetic material from F plus bacterium to the F minus bacterium, from donor bacterium to the recipient bacterium. Okay, so whenever, in a short I will explain once again. So whenever the recipient bacterium comes in contact with the donor, both comes close to each other, by the help of pili present in the donor bacterium attaches to the recipient and forms a tube-like structure, bridge-like structure, that is called as conjugation bridge, and the replicated the genetic material DNA will be passed into the recipient. So now recipient also has a plasmid, F plus plasmid. Okay, so hence F minus bacterium is now considered as F plus bacterium. Okay, so this method was first studied by Liderberg and Tatum. The second method, it is called as transformation. Okay, so here this method was first studied by Griffith. See here, transformation. Here we could find that a bacterium will be changed. Okay, so changing its form, it will be seen here. A bacterium What is transformation? Picking up of genes. Picking up of genes from its surrounding environment by a bacterium is called as transformation. Picking up of genes by a bacterium from its surrounding environment. Okay, who will give the genes and how this bacterium will pick the genes? See here, in the surrounding environment of this bacterium, if there is another dead bacterium is present, a dead bacterium is present, that dead bacterium will undergo disintegration. Okay, it starts disintegrating. Okay, it starts disintegrating, and this bacterium will pick up the genetic material from the dead bacterium, and that the genetic material of this bacterium will be incorporated into this bacterium. That process is called as transformation. Genetic recombination, mixing up of its own with the another bacterium genetic material. See here, transformation takes place only when the bacterium completes its growth. Okay, and it should have a capacity to pick up the genes from its surroundings. So it has to complete the growth. Then only it has achieved the capacity to take up the genes. So here, transformation means it is the process of picking up of the genes by a bacterium from its surrounding environment. So whenever in a surrounding environment there is a dead bacterium, and that the dead bacterium undergo disintegration, and the bacterium will take up the genes, and that will be fixed, incorporated into its genetic material. Okay, a new genetic material is being formed, genetic recombination is being seen here, and that process is called as transformation. And the third method, it is transduction. Transduction was first studied by Liederberg and Zinder. Liederberg and Zinder studied transduction. Transduction is a process of transfer of genetic material or the DNA from one bacterial cell to the another bacterial cell by the help of, okay, you can call it as a mediator or by the help of a vector. So it requires another organism for transferring of the genetic material from one bacterial cell to the another bacterial cell. When from first bacterial cell, the DNA or genetic material, it has to be transferred to the second via the help of mediator or another organism, or it might be the vector. So here vectors are nothing but the viruses. Viruses are being involved in transferring of genetic material. The viruses that will infect the bacteria, they are called as bacteriophage. The virus that will infect the bacterium, so they are called as bacteriophage. So bacteriophages are involved in transferring of the genetic material from one bacterium to the other several bacterium. That process is called as transduction. So this is a one bacterium. So this bacterium is being infected by the virus bacteriophage. Yes. So when this bacterium is being attacked by the virus, infected by the virus, the bacteria will be disintegrated. But what does the virus do? The virus will incorporate the genetic material. Okay, so we could find that the genetic material will be taken up by the virus. And the virus will produce many copies of new virus which has the genetic material of the bacterium. So now those viruses which have the genetic material of bacterium is ready to attack the other bacterium. Okay, so now that bacterial DNA genetic material will be introduced into the several bacteriums. See here, so how does transduction takes place? So the viruses will attack a bacterium, that is bacteriophage. What is bacteriophage? A virus that will particularly infect only the bacterium, they are called as bacteriophages. The bacteriophage will first attack the bacterium. Okay, so now when it attacks the bacterium will slowly be destroyed because it has been virus has been attacked to the bacterium, so it will be destroyed, disintegrated, but its genetic material will be incorporated, it will be taken up by the virus, and that virus produces many copies of its own. New viruses are being produced which is having the genetic material DNA of its own as well as bacterial genetic material. Okay, so now these bacteria, that is viruses, bacteriophages, are ready to infect the other bacterium. As they attack, and there we could find the new bacterial DNA will be introduced into the another bacterial cell. This process is called as transduction. Transfer of the genetic material from one bacterial cell to another bacterial cell by the help of virus, or it's also called as bacteriophage. Okay, so in case of a bacterium, we could find reproduction is by a sexual method, and sexual method which is also known as, it's a primitive method, so which is also called as parasexual or genetic recombination. Okay, so here mostly the in case of bacteria, we could find that a sexual method, it is the method of reproduction. Okay, so kingdom Monera is being divided into Archaebacteria and Eubacteria. In Eubacteria, so you have studied about the bacteria, its classification as well as reproduction. Okay, so another group in case of Eubacteria, it is Cyanobacteria and Mycoplasm, that we will study in the next class. Thank you.